All right, welcome to the deep dive. We're all about getting right to it here,
Yeah.
cutting through all the noise and giving you the knowledge you need without all the extra stuff.
Yeah.
You're here because you want to learn. You like those aha moments. You want a shortcut to really grasping complex ideas.
I like that, a shortcut
Yeah.
to knowledge.
Today, we're going to do something a bit unexpected.
Ooh.
We're going to dive deep into the world of microbiology.
Microbiology. Okay.
But we're going to do it as philosophers.
Right.
So you might be thinking, why should philosophy, a field often concerned with things like human existence, consciousness, really big questions.
Yeah, the big stuff.
Why should it concern itself with these tiny, tiny organisms?
It's a fair question. I mean, historically, even within the philosophy of biology, the focus has been mostly on animals, what we call the zoocentric perspective, right? A view that's centered on animals.
Right.
And although there's been, thankfully, a shift to include fungi and plants in recent years, microbes still tend to be left out of these philosophical discussions.
They're kind of on the sidelines, yeah.
But that's precisely where the potential for really surprising insights lies, I think.
Okay.
Our mission in this deep dive is to demonstrate why paying close attention to these minuscule, often overlooked organisms is not just some niche interest,
Right.
but actually a crucial element in both the philosophy of science and broader philosophical inquiry. And I think you'll find, as we have, that looking at life through this microbial lens can lead to some truly mind-bending revelations.
It's a whole new perspective.
Yeah.
So how are we going to approach this?
Okay, so we'll start by laying out the fundamental biological reasons why microbes are so important.
Okay.
Then we'll explore how they actually challenge some of our core ideas in the philosophy of biology, making us rethink those concepts.
Interesting.
And finally, we'll delve into the far-reaching implications of microbiological science, especially microbiome research, which you've probably heard a lot about,
Yes.
for a wide range of philosophical questions, including how we understand ourselves and our place in the world.
Big questions, yeah.
Yeah, the big question.
Okay, well, let's jump right in. Starting with the essential biological reasons why we cannot ignore the microbial world.
All right, let's do it.
So the first point is that humans exist in what is fundamentally a microbial world.
Okay.
This isn't just some catchy phrase. It's about the sheer ubiquity of microbes across the globe
Mm-hmm.
and the absolutely vital roles they play in every single ecosystem on Earth.
They're everywhere.
They really are.
Yeah.
And we can think about it in terms of their ecological functions
Okay.
and then also their immense evolutionary history.
Right. They've been around a lot longer than we have.
Yes.
So let's start with their ecological roles. When we talk about the planet working, we're essentially talking about these biogeochemical cycles. Think of them as the planet's, essential life support systems.
Yeah.
Constantly recycling crucial elements like carbon, oxygen, and nitrogen. And these cycles are all interconnected. And they've shown remarkable stability over vast stretches of geological time. Now, geological processes play a role over those immense stretches of time, of course. But the more rapid cycling of these elements, the stuff that sustains the world as we know it,
No.
that's largely driven by microbial activity.
So they're the ones making it all work.
They really are. Now, here's a key insight. Microbes, in their constant drive to extract energy from chemical compounds and even light, they are the real engines of these cycles. And often they dominate even when their total biomass is less than that of the larger organisms involved.
So even though they're small, their impact is huge.
Their impact is absolutely massive, yeah. Take, for instance, the carbon and oxygen cycles. Now, plants make up a large portion of living matter, but a significant chunk of that is, non-living wood.
Right.
Which isn't actively participating in these immediate cycles.
Yeah, it's just kind of there.
Right, it's just sort of hanging out. Now, the microbes, on the other hand, they're constantly at work, constantly churning things over.
They're busy.
They are. And what's fascinating is that microbes tend not to, hoard elements in their biomass for very long. They have this incredibly high turnover rate.
Right.
Their populations are constantly metabolizing and then being recycled. And this rapid cycling might be a key reason why we have these relatively fast biogeochemical cycles on Earth.
So they're keeping things moving.
Keeping things moving, preventing essential elements from getting locked away in long-term geological sinks. Now, here's a thought. We wouldn't even be here without them. I mean, think about oxygen.
Yeah, we need it.
We need it to survive, right? It's what allows our energy-hungry bodies to function. And where did all that oxygen come from? From microbes.
Yeah.
Specifically, these oxygen-producing microbes called cyanobacteria.
So they're the real heroes of this story.
Yeah. They're the original oxygen makers, yeah. Nearly 3 billion years ago, they started releasing oxygen as a byproduct of photosynthesis. And even more remarkably, their oxygen-producing machinery was eventually incorporated by later eukaryotes, leading to algae, plants, and ultimately the oxygen-rich atmosphere that allowed animals like us to evolve.
So in a way, our very existence is thanks to these ancient microbes.
Exactly. In a very real, if distant, sense, philosophy itself is built upon a foundation of bacterial metabolism.
Wow.
It's a humbling thought, isn't it?
It is. But their influence isn't just on a planetary scale, is it?
No, not
It's happening all around us, even inside us. We're talking about symbiosis.
Yes, this intimate living together of different organisms. Symbiosis is this fundamental organizing principle in the microbial world, but it extends far beyond microorganisms. It encompasses pretty much every relatively large living thing.
It's everywhere.
It's everywhere. Now, we often categorize these relationships based on their outcomes, right? So you have parasitic, which is harmful to one partner.
Right.
Mutualistic, which is beneficial to both partners.
Got it.
And then commensal, which is beneficial to one and neutral to the other.
Interesting.
Yeah, so examples. We see this all around us. Think about lichens, those like crusty things you see growing on rocks and trees. They look like a single organism,
They do.
but they're actually a tightly integrated partnership between a fungus and photosynthesizing microbes, either algae or cyanobacteria, or sometimes both.
Wow, so it's a team effort.
It's a team effort, yeah.
Yeah.
What's cool here is how these distinct organisms come together to form this new recognizable entity that can reproduce across generations.
So they become more than the sum of their parts.
Exactly. And generally it's believed that both partners benefit, leading to potentially increased reproductive success for both the fungus and the microbe.
A win-win situation.
Win-win situation. Then you have more manipulative symbioses like the Wolbachia bacteria in insects. This bacterium is often essential for the insect's reproduction, and it can even manipulate the insect's sex ratio, like killing males, all to ensure its own survival.
That's pretty sneaky.
Oh, it's a form of reproductive hijacking. But what's fascinating is that even in these cases, the insect host can sometimes gain advantages from the Wolbachia infection. It's a complex dance of manipulation and potential benefit.
So sometimes even a seemingly parasitic relationship can have benefits.
Exactly. It highlights the interconnectedness of life and how these relationships can evolve in really surprising ways. And then, of course, there's us. We humans are walking, talking ecosystems teeming with microbes.
I knew you were going to say that.
Mostly in our gut, but also on our skin, in our lungs, and basically every bodily opening.
We're covered in them.
We are. The vast majority of these microbes in our gut are just there, taking advantage of the abundant nutrients. Our immune system is constantly working to keep them in check so they don't become harmful, but we can't entirely eliminate them. This inescapable symbiotic relationship means that our bodies aren't just human. They are fundamentally and inseparably microbial ecosystems. And we'll explore the philosophical implications of this a bit later.
I'm looking forward to that. But before we move on, we have to mention the Gaia hypothesis.
Ah, yes. This compelling idea that the Earth itself functions as a kind of self-regulating system where life actively maintains conditions that are favorable for life.
It's a pretty radical concept.
It is. What's particularly relevant about the Gaia hypothesis, at least in its original form, is that it places primary responsibility for the mechanistic regulation of this system on microorganisms. All life participates, but the sheer abundance, metabolic versatility, and pervasiveness of microbes put them in this pivotal role.
So they're the key players.
It's a controversial hypothesis, no doubt, but it really powerfully highlights the ecological significance we must attribute to microbes, because they're fundamentally shaping how life operates on Earth.
Okay, so ecologically, they're vital.
Vital!
But their importance stretches back much, much further. Let's dive into those evolutionary reasons why we need to pay attention to them.
Okay, time travel. Think about the immense timescale of life on Earth. The paleontologist Stephen Jay Gould famously pointed out that the most striking feature of life has been the stability of its bacterial mode from the very beginning of the fossil record, which is about 3.8 billion years ago, right up to the present day.
It's a long time.
It is. He called this the age of bacteria, and by bacteria, he likely meant it in a broad sense to include archaea.
Right.
So life originated, as we said, about 3.8 billion years ago. The first cells were almost certainly microbial, most likely bacterial. And for the next 2 billion years, life was exclusively microbial. Bacteria and archaea were the only inhabitants actively shaping the planet's chemistry, sometimes to their own detriment, such as, the rise of oxygen from cyanobacteria causing extinctions for those that couldn't adapt.
That's a bit ironic.
It is. But then later in evolutionary history, eukaryotic microbes emerged, eventually leading to the multicellular groups we recognize today, the plants, fungi, and animals, us. But even during all that diversification in the last 500 million years, microbial activity has continued unabated. So Gould's emphasis on this enduring age of bacteria really underscores the profound historical dominance and stability of microbial life.
Okay, so we've established that biologists have very good reasons to be fascinated by microbes. But now, let's get to the heart of our philosophical inquiry today. Why should philosophers care? Especially those who might not primarily focus on biology.
Right, because it's not immediately obvious, is it? Well, there are numerous ongoing debates within the philosophy of biology where neglecting microbes would actually seriously weaken or skew any conclusions you might draw. And we're going to focus on 3 interconnected areas where microbes force us to fundamentally reconsider some core concepts: species concepts, the tree of life, and major transitions in evolution.
Those are big ones.
They are the big ones, yeah. Let's start with species concepts. This might seem a bit specialized, but it really touches on how we categorize and understand the incredible diversity of life. For animals, the biological species concept, the BSC, which is the idea that a species is a group of organisms that can interbreed within itself but not with others,
Right.
that seems to work reasonably well,
For the most part, yeah.
even though there are exceptions, of course. But when you start looking beyond animals, things get more complex.
Things get messy.
Plants introduce some challenges, but many zoologists think these are still manageable within a broadly BSC framework. Fungi, however, with their diverse and often bizarre reproductive strategies, they really start to stretch the limits of any universal species concept. And then, historically, there's been a tendency to simply dismiss asexual organisms, like many microbes, as not even forming species in the first place.
So they're not even in the game.
Not even in the game. And that brings us to bacteria and archaea. They really throw a curveball into our traditional understanding of species, don't they? No sexual reproduction, for the most part, very limited physical forms or morphologies, and no clear reproductively isolated lineages.
Right, it's all very different from what we see in animals.
Very different. And to make things even more challenging, there's this widespread phenomenon of horizontal gene transfer. Think of it as gene swapping between unrelated microbes.
They're just trading genes.
They're just sharing. And they do this with surprising frequency, which confounds any straightforward lineage-based view of species.
Yeah, it really shakes things up.
It shakes things up. So when philosophers start to seriously consider microbes, especially bacteria and archaea, they're almost immediately pushed towards a more pluralistic view of species concepts. What works for sexually reproducing animals simply doesn't apply across the entire spectrum of life.
So there's no one-size-fits-all definition of a species.
Not at all. It compelled us to acknowledge that there isn't likely to be one single universal definition of what constitutes a species.
So microbes force us to rethink these fundamental categories.
Exactly. And that leads us directly to our next point, the tree of life. We often visualize the relationships between living things as this branching tree, with each branch representing lineages diverging over time. Traditionally, these phylogenies were built using observable physical characteristics, including fossils. But the arrival of molecular data gene and protein sequences has given us this much more detailed view, especially for organisms with limited physical forms like microbes.
The molecular data gives us a clearer picture.
It does in many ways, but it's also revealed something quite unexpected. That there's a substantial amount of gene exchange occurring between prokaryotic lineages, even those that we think of as defining major branches of the tree.
So the branches are all tangled up.
They are. This widespread horizontal gene transfer, as it's called, really blurs those neat vertical lines of descent that the traditional tree model relies on. We also see hybridization in multicellular organisms, which complicates things. But the sheer scale of gene swapping in the microbial world makes the standard tree of life, as a simple graph of purely vertical inheritance, really questionable, conceptually and practically.
So it's not as straightforward as we thought.
Not at all. So any philosopher contemplating what the tree of life actually represents has to grapple with the reality of microbial evolution.
It changes the whole picture.
It really does.
Yeah.
And this takes us to major transitions in evolution. These are pivotal events in the history of life. such as the origin of cells themselves, the evolution of sex, or even the development of language in humans.
These are like the big turning points.
The big moments, yeah. Now, philosophers who study these transitions, they often try to find a unifying theme, some common thread that links these seemingly very different events. And a recurring suggestion has been the concept of individuality, that major transitions involve a fundamental shift in what we consider to be an evolutionary individual.
Okay, so how do microbes fit into all of this?
Well, that's where it gets really interesting. Microbial evolution is absolutely fundamental to understanding these transformations, even if your primary interest is in eukaryotes, which is the domain encompassing plants, fungi, and animals, or even just metazoans, the animal kingdom that includes us. Take the very origin of eukaryotes. The traditional view was that they arose from a symbiotic merger of a bacterium and an archaeon, forming a distinct third domain of life.
So two became one.
Right, this fusion idea. And this was partly based on the apparent complexity and diversity of eukaryotes, all the different forms they take. But recent discoveries of a diverse group of archaea called the Asgard clade have really changed this picture. Genetic analysis of these Asgard archaea has shown they have genes that were previously thought to be exclusively found in eukaryotes.
Wow, so they share some key genes.
They do. This suggests that eukaryotes didn't just arise alongside archaea, but actually within the archaeal domain. It now seems like the origin of eukaryotes was a more gradual process of gene diversification, potentially including gene transfers, and that the acquisition of the mitochondrion, which is a key feature of eukaryotic cells, might have happened later in the process.
So the story's more complicated than we thought.
It's more nuanced. It shifts the focus away from a single dramatic individuality transition and more towards a story of gradual diversification within archaea. Meaning, to truly understand our own origins as eukaryotes, we need to understand the evolution of these ancient microbes.
You can't ignore them.
We can't. And a similar pattern is emerging when we look at the origin of metazoans, the multicellular animals. While it's not always classified as a major transition in the same way as the origin of cells, it's obviously crucial for understanding human evolution. And recent progress in this area has relied heavily on studying the unicellular relatives of animals, things like choanoflagellates, philisterians, and ichthyosporians.
So we're looking at our distant single-celled cousins.
Exactly. And what's remarkable is that genomic and experimental analyses show that these unicellular organisms have the genetic toolkit for features that were once believed to be unique to metazoans.
All kind of features.
Things like cell adhesion, developmental signaling pathways, and specific gene regulatory factors. Even more surprisingly, some of these unicellular relatives can form sophisticated multicellular arrangements, sometimes even triggered by interactions with bacteria.
That's amazing.
It is. The emerging theme here is clear. Understanding even the evolution of complex multicellular life demands a microbial perspective.
Okay, so we've looked at microbes from the perspectives of biological classification, ecology, and evolution. But now let's turn our attention to the science of microbiology itself. It has a fascinating history and philosophical implications too, right?
It absolutely does. Carl Woese, a true visionary in the field, even proposed that microbiology would become the primary biological science. Now, that might be a bold statement, but there's no denying the profound impact of microbiology on our understanding of life.
So how did it all begin?
Well, the story starts with early microscopy back in the 17th century. This allowed us to see microbes for the first time. But the real transformation of microbiology into a rigorous scientific discipline came with its experimental turn in the 19th century. The focus shifted from just observing these tiny creatures to understanding their specific effects, particularly in relation to disease.
So it became more about figuring out what they do.
Exactly. And a crucial development during this era was the development of techniques like pure culture, where you could isolate and grow single microbial species in the lab.
So you could study them in isolation.
Precisely. This was essential for moving beyond simply observing microbes in these complex environmental samples to actually understanding their individual roles, especially in causing diseases.
And this led to Koch's postulates, right?
Yes. Think of them as a systematic method for rigorously establishing a causal link between a specific microbe and a specific disease. The process involves isolating the microbe from a diseased host, growing it in pure culture, introducing it to a healthy host to see if the same disease develops, and then re-isolating the identical microbe from the newly diseased host.
So it's about proving that this particular microbe causes this particular disease.
Exactly. It's a rigorous method for proving cause and effect.
And this led to the germ theory of disease, right?
Yes. Once you could reliably establish these specific and stable causal relationships, the germ theory of disease took hold. It provided a powerful predictive framework for understanding and explaining diseases.
So, for example, knowing that tuberculosis is always caused by a particular bacterium.
Exactly. The systematic and experimental nature of germ theory revolutionized medicine, transforming it from a largely observational practice to an increasingly scientific and evidence-based field with real therapeutic and preventative successes.
It was a game changer.
It really was. As a result, the study of microbes became deeply integrated into universities, hospitals, and even industry. And the impact of microbiology didn't stop there. It continued to transform other fields, leading to the rise of molecular microbiology in the mid-20th century.
So microbiology was branching out.
It was. Building on the successes of germ theory, microbial biochemistry flourished as researchers delved into the precise molecular pathways by which microbes exert their effects. The relative ease of working with microbes in the lab, combined with their incredible metabolic versatility, led to a vast amount of research in bacterial biochemistry, and this laid the crucial foundation for the emergence of molecular biology in the 1940s, where many of the initial major breakthroughs were made using microbial systems.
And wasn't E. coli a key player in all of this?
Absolutely. E. coli became this, undisputed superstar of molecular biology. It was the go-to model organism for understanding fundamental processes of life: gene structure, replication, and expression.
So it was the model organism for understanding life at its most basic level.
Exactly. E. coli and other prokaryotes provided us with the very language and much of the fundamental knowledge of molecular biology that we still use today, and it's relevant to all organisms. Even the groundbreaking discovery that DNA is the hereditary material was made using bacterial and viral systems.
Wow. So microbes were at the heart of some of the biggest discoveries in biology.
They really were. And then, as technology advanced, we saw the expansion into genome sequencing and metagenomics in the microbial world, opening up entirely new avenues of research.
So we could start to look at the bigger picture.
Exactly. What's remarkable is that this broader molecular perspective not only led to new technologies, but also provided these major evolutionary and ecological insights.
Into the history of life.
Yeah. And the dynamics of microbial communities across the planet. As we talked about earlier, much of our current understanding of major evolutionary transitions comes directly from these microbial genomic investigations.
So both in its early stages and in its more recent developments, microbiology has heavily relied on using microbes as these general molecular models for understanding life and driving technological innovation.
They're incredibly powerful tools. Their tractability in the lab and their metabolic flexibility have made them invaluable. Consider the E. coli operon model of gene regulation. Even though eukaryotic gene regulation is significantly more complex, this operon model launched an entire field of investigation based on this fundamental principle of how gene expression can be controlled.
So microbes are great for understanding molecular processes, but their value as models extends beyond just the molecular level, doesn't it?
Absolutely. Even acknowledging that no model system is perfect, microbes have proven to be remarkable models for studying living processes more generally. In the life sciences, we use what are known as model organisms. These are species that are studied extensively in the lab.
Like fruit flies or mice.
Exactly. Nematodes, fruit flies, mice, yeast, things like that. And microbes are often considered particularly tractable model systems, especially for studying evolution and ecology in action.
Now, here's a point where philosophy of science comes in, right? Because philosophers have actually dedicated a lot of thought to the nature of model organisms. Are they primarily analog models, like, say, a wind tunnel model of an airplane?
Yeah.
Or are they more like representative specimens from which we can extrapolate to broader groups of organisms?
That's a great question, and there's ongoing debate about this, of course, but there is a general consensus that microbes often act as analog systems in experimental settings, especially in experimental evolution and ecology.
Can you give us an example of that?
Sure. Think back to Jordi Goss's pioneering work in the 1930s, where he used paramecia and bacteria to model interspecies competition. He wanted a simple laboratory system to understand the fundamental mechanisms of this struggle for existence that Darwin talked about.
Right.
So he set up these populations of two species of paramecium that both consumed bacteria. And he observed that when they competed for the same limited bacterial resources, one species consistently drove the other to extinction. And this was precisely as predicted by the logistic equation.
So the model accurately predicted what would happen.
Exactly. And this work led to the competitive exclusion principle, which is a cornerstone of ecological theory. But he also tried to model predator-prey relationships using microbes.
And did that work?
Not quite as well. He used a paramecium and its unicellular predator. But instead of observing these oscillating population dynamics that are predicted by the Lotka-Volterra equations, he often found that both populations crashed to extinction.
Oh, so the model didn't quite capture the complexity of the real system.
Exactly. This highlighted that even seemingly simple microbial systems might not perfectly capture the complexities of the real world.
Right.
And it actually took decades of further research with various organisms before those predicted oscillations were reliably observed in a lab setting. Even then, it often required very careful manipulation of the system.
So even with microbes, modeling can be tricky.
It can be, but the limitations of some models don't diminish the power of others. Consider Richard Lenski's long-term evolution experiment with E. coli. It's been running continuously since 1988.
That's an incredibly long experiment.
It's an iconic example of experimental evolution in action. Lenski and his team started with 12 genetically identical populations of E. coli in identical controlled environments. And they've been tracking their evolutionary changes for over 70,000 generations.
So they're watching evolution unfold in real time.
They are. And because the starting conditions were the same, any differences that arise in these populations are due to spontaneous mutations within each of those lineages.
So it's all about random mutations driving evolutionary change.
Exactly. And they've gained remarkable insights into the long-term dynamics of adaptation and diversification, the significant role of chance in evolution, and even the emergence of genuinely novel biological features. Well, for example, one of the E. coli lineages evolved the ability to use citrate as an energy source in the presence of oxygen. This was something the ancestral strain couldn't do. And by studying frozen samples of the bacteria taken at various points throughout the experiment, they were able to reconstruct the entire evolutionary trajectory of this novel trait.
They could trace the steps of evolution.
They could, which brings us to the Avita digital evolution platform. This was inspired by the E. coli experiment, and it allows researchers to study evolution in a virtual world.
So it's like a computer simulation of evolution.
Exactly. Digital organisms evolve and compete for resources within this computer program.
Yeah.
And the aim, just like with the E. coli experiment, is to uncover those fundamental principles that govern evolving systems.
So it's a different way of studying the same basic processes.
Precisely. And they can actually compare the results from these virtual experiments with the results from the real-world E. coli experiment, which is fascinating.
And this whole idea of having a frozen fossil record is really powerful. Being able to go back in time and revive ancestral populations to see how they differ from their evolved descendants gives us this incredible window into the process of evolutionary change.
It does. And this ability to back up and rerun evolutionary trajectories in microbial experiments has some interesting parallels to what happens in computer simulations, and even to Stephen Jay Gould's famous thought experiment about replaying the tape of life.
Right, the idea that we could rewind and replay evolution. Would things turn out the same way?
Exactly. While Gould was thinking about the entirety of life's history, these microbial experiments demonstrate that we can actually replay certain aspects of life's evolutionary history in the lab.
That's amazing. And it raises some really interesting philosophical questions about the nature of modeling and how we use models to understand the world.
It does. I mean, it's fascinating to compare these concrete microbial models to the more abstract mental models that philosophers often use in their thought experiments.
So microbes are helping us to rethink how we approach philosophical inquiry.
They are. Now, while philosophers studying really complex phenomena like cognition might naturally gravitate towards animal models for investigating these core life processes, microbes offer this level of experimental tractability that can be incredibly insightful.
Okay, so microbes are clearly powerful models, but are there inherent limitations to what they can teach us? Aren't there many complex animal behaviors and phenomena that just don't have any meaningful microbial counterparts?
That's a valid question. There's a natural skepticism that many complex human and animal phenomena might be too far removed from anything we observe in the microbial world to allow for useful comparisons. However, it turns out that microbes are actually proving to be surprisingly productive study objects for phenomena that were initially defined by animal characteristics.
Really, like,
Well, take predation, for example. We typically think of predation as one animal actively hunting and consuming another animal.
right, like a lion chasing a zebra.
Exactly. But the scientific definition of predation has actually broadened considerably. And we now recognize that bacteria also prey on other microbes.
They do.
They do. Early discoveries highlighted bacteria that explicitly attack and consume other bacteria cells. And today we know there's this diverse range of predatory behaviors within the microbial world. And for many research purposes, these bacterial predator-prey systems can serve as valuable models for understanding general predation dynamics and its ecological impacts, even revealing limitations in existing ecological theories, as Gauss's early work illustrated.
So even though they're tiny, they can teach us about these big ecological concepts.
Exactly. Another example is aging. You might think, surely unicellular organisms don't experience aging in the same way that multicellular organisms do.
Right, they just divide and create new cells.
That was the prevailing view for a long time. Unicellular life was considered ageless because cell division was thought to be symmetrical, with new offspring essentially replacing the parent cell. But there is now a growing body of research that's investigating aging in bacteria, carefully distinguishing between death and the process of aging in these organisms, and exploring what this means for our understanding of the evolution of senescence more generally.
So even single-celled organisms age.
It seems so, and this means that bacterial aging can actually be a model for core aspects of the process of growing old.
That's really interesting. What about something like kin recognition? That seems very intrinsically linked to animal sensory and behavioral abilities.
Initially, yes, but we now know that kin recognition, based on chemical sensing and interaction, does occur in microorganisms as well. And what's particularly interesting is that in bacteria, the underlying mechanisms are biochemical, often allowing for this relatively straightforward and detailed analysis compared to the complexities of behavior in animals.
So we can study it in a more controlled way?
Exactly. Researchers studying protists even argue that unicellular eukaryotes are ideal model systems for investigating the origins and evolution of kin recognition. These are simplified representations, of course, but that's inherent to the practice of scientific modeling.
Right, we're always simplifying things to some extent. Now, the big one, cognition. Can microbes really teach us anything about cognition?
That might seem like the biggest leap, right? From single-celled organisms to brains and minds. But unicellular organisms, both eukaryotic and prokaryotic, have been extensively studied for capacities such as decision optimization, efficiency and resource use, memory of past conditions, anticipation of future states, and even basic forms of self-awareness in response to their environment.
So they're not as simple as we might think.
Not at all. Now, the argument isn't that microbes possess the same cognitive abilities as animals, but rather that they exhibit capacities that share fundamental aspects with cognition and can be studied as valuable analogs for understanding cognition more generally, including in animals.
So by studying microbes, we might gain a better understanding of what cognition is and how it works.
Exactly. And the ongoing debates around minimal cognition and this emerging view of cognition as a diversely distributed capacity across the tree of life really highlights how examining microbial research can challenge and enrich our philosophical thinking about classic subjects like cognition. And this leads us nicely into our next major area, the microbiome.
The microbiome. This is a huge area of research right now, focusing on the communities of microbes that live in and on host organisms. And there's particular interest in the human gut microbiome and its links to physical health, disease, and even mental well-being.
Absolutely. And from a philosophical standpoint, microbiome research raises some profoundly interesting questions.